tree project is mainly about effects, long-term effects of radiation on wildlife under natural conditions, using the Chernobyl Exclusion Zone as a natural laboratory. But there is also an element of long-term biogeochemistry of those radionuclides, because the Chernobyl Exclusion Zone provides us with a perfect natural open laboratory for testing the long-term behaviour of some of the nuclides that were ejected from the reactor in 1986, because obviously we're now 29 years after that accident. So that's the part, that sort of element of the work is the part that that University of Nottingham is involved with. Right. Okay. So this is a forest. It's a very small piece of forest. The tree species is Scots pine, and the reason for selecting this, it's the best sort of what we call a parcel of Scots pine that we could find near Burakovka. So we are looking at actually 4 specific elements, or radioactive nuclides of elements, including uranium technetium, which is an artificial element and doesn't actually exist in nature. Actually, Chernobyl is one of the very few places in the world where you can find technetium actually out in the open environment. So the uranium and technetium are 2 of the elements. The third one is iodine, which is a very important element. One of the isotopes of iodine, iodine-131, was very, very important in terms of the radiological impact of the accident. We're actually looking at iodine-129, which has a much, much longer half-life than iodine-131, so it persists in the environment much longer. And the 4th element is selenium, which actually, there is a radioactive isotope of selenium called selenium-79, which actually wasn't ejected from the reactor as far as we know, but is an important isotope in nuclear waste. And that's just the 4th of the group of elements that we're looking at. Obviously, we've been driving along the roads past some really big areas of Scots pine, and it's a very, very important, was a very, very important commercial species in the zone. It's planted, and if you look back here and look along the line, you'll actually see that it's planted in straight lines, which gives away the game. So it's not growing naturally. It's planted. So all the trees we tend to see in a forest of pine would be roughly the same, well, probably exactly the same age actually, apart from a few individuals that might have seeded themselves. So, as I say, it's an important commercial species. The other reason for looking at a Scots pine plantation to do this forestry exercise is that Scots pine is the most, as far as we know, the most radiosensitive species of any animal and any plant. Obviously there's a lot of dose effects that we don't know about that may have been going on with organisms that are more difficult to see and to measure. But it was the Scots pine which were killed within the Red Forest by the very, very high dose rates immediately after the accident. No other trees, as far as I know, were killed on a large scale. So we know that Scots pine is quite sensitive to high radiation. So that's another interesting reason for looking at it. And of course, the Scots pine that was killed in the Red Forest became quite a waste disposal problem. And one of the things that we might be seeing, I think, I'm not sure whether it's still scheduled, but was to go and look at one of the waste disposal trenches near the Red Forest. When the trees died in the Red Forest, pine trees, they were bulldozed, and they were bulldozed into trenches just to get rid of the waste, and it became radioactive waste. So again, understanding just how much radioactivity the trees can hold is an important part of the kind of radioactive waste story. So one of the reasons the Red Forest got so much radioactivity in the initial phases of the accident is because tree canopies generally, but coniferous canopies in particular, are very, very effective at trapping small aerosol particles from the atmosphere. I've actually had the chance to revisit one of my old field sites, which I last came to in 1994. So I've not been here for a long time, about 21 years. And it was one of a series of forest sites that I sampled back then. I was working with the University of Moscow in the early 90s, and we had a series of forest sites here and elsewhere across Europe. Obviously some of the forest sites here are particularly interesting because of their relatively high, well, their high levels of contamination. So I had a chance to go back with one of my current PhD students and resample one of those sites. So we're going to be looking at the change, if any, in the distribution of cesium in the soil profile since 1994 and present day. One of the reasons for choosing Burakovka is it's on the trace, the plume of radioactivity that came out of the reactor during the initial phase of the accident, and it's long and thin, and it's very, very high levels of deposition in the middle, and we're kind of on the edge of the western trace here at Burakovka, so that's why the dose rates are reasonably high. In that western trace there was a mixture of particle sizes. Some of the particles were quite large hot particles, so between 10 and 100 microns in diameter, which is a large particle in terms of what we find in the atmosphere. So they will fall out of the atmosphere pretty quickly. So we tend to find the larger particles closer to the reactor, and the further away from the reactor we get, the smaller and smaller the particles become, until we get thousands of kilometres downwind, and we're only picking up the very, very small 1 micron particles that can remain suspended in the atmosphere for many, many days and weeks. So we will have had a mixture of large and small particles deposited here. The trees are particularly good at filtering out from the atmosphere the small particles in particular. Okay, so we would expect to find higher levels of deposition in a tree canopy, in a woodland, in other words, than in the neighbouring grassland. We've also been picking up samples for our work with the tree project. With the students specifically, we've been looking at some of the, I've been introducing them to some of the more general elements of forest radioecology, looking at the things that control the cycling of radiocesium, in particular around the forest, and looking at some of the components of the forest using various methods like soil coring, tree coring, and in situ methods of estimating biomass of forests. Right, so what are we going to do here? We're going to try and work out, we'll be sort of learning a little bit about the structure of the place, what the soil looks like, and and other aspects of the forest. But we're going to try and work out where the radioactivity is. Okay. Craig is going to actually do we've done a dose survey here already. Actually, not a dose survey, a radioactivity survey. And so from those data, we should be able to determine an average deposition value, an average becquerel per metre squared value for the forest floor. And of course, some of that radioactivity is in the trees. Some of that radioactivity is in the soil. We'd like to get an idea of just what those relative distributions are, and also we might want to calculate where, you know, other components of the ecosystem, where the radioactivity is. You know, we've got animals walking around carrying radioactivity. How much radioactivity are they carrying? We get seasonal growth of mushrooms, for instance. How much radioactivity is in the mushrooms? We're going to just concentrate on the trees and the soil because they're the major structural components of the forest. So the first thing we need to do is to work out what the standing biomass is. So we're going to set out basically a big quadrat using this tape measure, it's a 60 metre tape measure, and the first thing to do is to set out a big square. So we'll set out a square along, you know, using the trees, some of the trees as corners. We're going to count the number of trees in that big square of known area. Okay. And then the second thing is we're going to try and estimate using two very, very easy measures of the trees, just how much standing biomass we've got. First thing we need to know is the height of the trees. Right, so I'm going to ask you to shin up the tree with a tape measure all the way to... No, I'm not. So there is another way of doing it. Okay, we're going to be using a clinometer, which is a means of measuring the angle from you to the top of the tree. And if you measure that, if you can look along a 45-degree angle to the top of the tree, and then measure the distance from you to the tree, and then add the height of your eye, then that's a pretty good approximation of the height of the tree. So we're not going to ask you to shin up the tree, because that's not in the risk assessment. Second thing is, that's one basic measurement. The second basic measurement is to measure the circumference of the tree at chest height. Okay, standard forest measurement. It's called DBH, diameter at breast height. So it's difficult to measure the diameter accurately. So what we do is we measure the circumference, and then with a bit of arithmetic using pi, we can calculate the diameter. All right. So we're going to do measurements of 6 tree heights, reasonably randomly, 6 DVHs reasonably randomly, and from that we can work out the standing biomass. The forests are actually very, very important in the zone. They currently occupy something like 70% of the total area of the exclusion zone, so they're pretty important part of what we see around us. Perhaps you can choose a tree at random over there, and if somebody then wants to run the tape measure along to another tree over there, just marking out a square area. You've got to note down the dimensions of the area as well. So literally you just stand there, if that's one corner of the survey area, that tree. So if you want to take the tape measure and run it along to one of these trees here. Okay, let's just pass it round one of the trees. Use it, and then you can walk down in that direction. The best way to do this is one person goes to one corner and another person starts in the opposite corner, and then if you walk down slowly looking at each other, hang on, come back to the corner, you're going away from the corner there. You need to stand outside the survey area. Alright, and then just walk down slowly together. So if you want to stand just outside the survey area, between you, just count it quietly to yourself. Okay, and then when you get to the end, just compare counts and see how many trees you've got in the area.
Do you mind if I preempt the recording spot so that we can have the circle?
Okay, so the next thing is to we've got 3 clinometers. So the idea here is to look up at the top of one of the trees. We want to just randomly select 6 trees. These are just very simple devices which allow you to measure angle. So we want to view the top of a tree at 45 degrees. Okay, so to view the top of the top of any particular tree at exactly 45 degrees, you've kind of got to adjust where you are, your distance from it. All right. So if you adjust your distance so that when you're looking, you're looking at 45 degrees at the top of the tree, you can then measure the distance from you to the tree, add the height of your eye, and then that's a pretty good approximation of the height of the tree. Alright, so there are 3 of these. It's quite easy or useful to do these in pairs because of the design of this clinometer. You can't actually see the angle when you're taking when you're sighting. So somebody can look and say, yeah, that's 45 degrees, no, it's only 40 or it's only 30. Okay?
Oh, I need to come on the other side, that's almost right.
Is it?
Yeah, that's 45. Oh, wow.
Yeah, so that's stretched to dry level. Right, if you run the tape measure out to the tree, and then bingo, you've got the...
Which tree?
The one, keep what? If you keep
He'll tell you where to go.
Yeah. To your left slightly. Keep walk, straight ahead of you.
That one.
To your right now. Yeah, that one.
Yeah. So basically, if you've got that measurement right, if that tree were to be cut and fall, it would just miss you.
Yeah.
Yeah.
Climbing a tree is if you're trying to measure the height of one particular tree in the middle of a dense forest, the clinometer, you might find it tricky with a clinometer. Do you know what I mean?
Yes. Oh yeah, sure. Yeah. Yeah. We've got a measure of the total mass of timber, or we will have a measure of the total mass of timber that's standing here. It would be nice also to get a sample of wood, so we could do some analysis on the chemistry of the wood, so cesium content, nutrient content, etc. We could also look at the growth history of the trees, if we could get a sample of the wood. And obviously, the easiest way to get, the most obvious way to get a sample of the wood is to cut a tree down, but that's a big job. First thing, and secondly, we don't want to necessarily go around chopping down lots of trees, even though it's in an abandoned zone. So what we'll do, we'll take a tree core, and I'm going to take this one over here. So basically, it normally goes in about sort of breast height, and it's really quite a sharp cutting edge on the screw, so it should go in really easily. You can get these in different sizes. I mean, some of these tree corers are very, very long. You can get them about sort of 2 feet, 2'6" long, and you can sample. I mean, the dendrochronologists use these to sample trees that are maybe hundreds of years old. You can also use these in old timbers in buildings and ships. So if you have historic ships and buildings, you can take samples from old timbers. And the dendrochronologists have this continuous record going back from the sort of present day, back sort of hundreds of years. And so if you get a piece of timber from an archaeological dig, for instance, you can actually, a bit like the DNA sequence, you can actually sort of slide the thing along till it matches, and then you can say, oh, it came from that period of 1300s or 1400s. So we can basically count back, we can, count the rings back. If we count back 29 rings, that should take us back to 1986. So if you want to hold that very carefully, without dropping the tree core, what I've got somewhere in my pocket, I have a hand lens. Okay. So it's much easier to do this under a low power microscope. But basically what we can try and do, this is very approximate, but we can try and count back 29 years. That's 1, 2. That there is 29 years. So it looks actually like these trees were pretty small when the accident happened, because you could see we've gone through the centre of the tree there. So I would say these trees are actually much older than, they must have been quite young when the accident happened. And what we've done is we've collected tree cores early this week from the Red Forest, from the sublethal area of the Red Forest. And what we're looking for is any, in that particular area where the doses were really high, and we've literally collected trees on the edge of the sublethal zone, so literally a few meters from where we've collected tree rings, trees, just a few meters away were being killed because the dose rate was so high. What we're looking for in those trees is any abnormalities in the growth rate. And I guess what we're looking for is kind of compression of growth. So you look to see smaller growth rings and reduced growth rate. But we haven't done that analysis yet. But that's what we're looking for. But anyway, so that wood was being formed when the accident was happening. Mm-hmm. Okay, so tree cores give us that sort of historic record, and we can either we could just chop that core up, look at the total cesium content, put it on a gamma detector. We'd very, very easily see the cesium-137 on a germanium detector in the lab. We might look at the time history. We know that's so the last 5 years, 5 to 10 years, 10 to 15, etc. So we might cut it into chunks and look at sort of the contamination over the recent history. The problem with that is that cesium moves around a lot. So the cesium that we measure in the ring in 1986, the ring that we know was formed in 1986, isn't necessarily the cesium that was put there in 1986, so it might have come in afterwards. So cesium's a very, very mobile element inside plants and trees. For some elements like lead, which are a lot less mobile than cesium, it works really well. We've done a similar analysis of growth rate for trees which were subjected to chronic acid rain during the 1980s. And there you can see that's on the Czech-German border where you got this process of forest death as a result of acid rain, and there you can clearly see the tree rings are compressed dramatically, and the growth rate of the tree dips completely in inverse relationship with the recorded sulfur dioxide concentration in the atmosphere. So it works for that. So we're going to see if it works for radiation from the Chernobyl accident, which we've not checked before. Okay. If you're going to help me, you need to put a mask and gloves on. And this is just because most of the activity, I'm kind of giving the answer away to the little calculation that we're going to be doing, but most of the activity is in the soil. A lot of it's near the surface of the soil still. And obviously if we're working, it's very dry, so if we're working with fairly active soil particles, we don't want to inhale them, and we don't want to take them home on our fingers basically. So we'll need that. We'll need a knife, and we'll need a corer. So this is Sergey's loaned me a couple of corers. I've tried this one and not had much success with it. So we're going to use this. It's just a tube, stainless steel tube with a sort of screw piston in it, and a couple of cow horns. If you all just want to sort of have a fairly close look at what we're going to be sampling, obviously the forest floor is covered in litter, forest litter, so that's twigs, and it's bits of, well, we've got birch leaf blown in from somewhere. Let's just take out a bit of grass there, which we'll just remove. Cones and needles. Okay, so this is the kind of rain of organic material that comes from the canopy, and in an evergreen canopy like this will come more or less all year. It may come in sort of, you may get more organic matter dropping at one time of the year than another. Perhaps it coincides with windy weather or dry weather when you get more needle fall. But basically this is the supply of organic matter to the soil, and we can just clear away just the litter very gently, and as we clear away the litter, we get to a point in the soil where we see it's kind of darker, and we see the sort of half decomposed remains or the semi-decomposed remains of the litter, and this is the fermentation horizon. So this is where the majority of the decomposition's happening. Just below that, if we dug down a little bit more, we'd come to what's called the humified layer. So the forest litter, in inverted commas, is actually composed of three discrete layers. There's the litter layer itself, there's the fermentation layer, and the humified layer. So what a lot of people do when they're sampling soils, soil core samples, is just get rid of all of that stuff and sample the mineral material underneath. But actually what they've done is to remove a really important part of the forest. That litter layer and all of the organic layers beneath it, that's really the very, very biologically active part of the system, and it's where a lot of the nutrient turnover takes place. Okay, and that's where a lot of the cesium is sitting. At least it was 10 years after the accident. I haven't seen recent depth profiles, so I can't tell you exactly where the cesium is. But we could determine exactly where the cesium is by taking a core and measuring what's in the core. So this is a fairly straightforward process. You just press this in, and because we've got nice sandy soils here, it should go in relatively easily. He said it probably not going to go in all the way. It's actually a 20 centimetre core of this one. Let's see if we can get it as deep as possible. I would say that's pretty good. Okay, so we can now just, if we have that handy, what we're going to do is pull it out, and we're going to turn the screw and we're going to sample approximate 5 centimetre depths.
George,
Oh, okay.
can you buy them as they are, or do you need? Do you need to?
A license to buy.
No, I mean I know that many of these devices, they are kind of homemade.
The tree corers, no, they're commercially made.
Right.
There's a huge industry in all this forestry measurement stuff. It's amazing what you can buy. Yeah. That's right. Wish me luck, because sometimes this doesn't work. Oh, it has. Hey, okay, that's actually a really nice one. So you can see how sandy. That's 20 centimetres down from the surface. It's very, very sandy subsoil. So what we're going to do, we're going to extrude about 5 centimetres at a time. I'm just going to turn this. This is difficult because it falls to pieces as soon as you press it out. So this is going to be quite approximate. Having said that, this is looking very nice. So, haha, how many centimetres? 2 centimetres, do you reckon? Approximately? Yeah. Let's say 2. Yes. Okay, so I need 3 more. Okay. Need 3 more. If I can see the tape. Need to see the scale of the tape. Huh? I'm going to say that's another 3. So if I can have the knife, please. Thanks. So we just trim that off, and what we'll do, we'll put that in a polythene sampling bag, which we've got over here. So if I hold that bag open, Kirill, can you just sort of pour that in fairly carefully? Oh, yeah, no, okay. But we need all of the sample because one of the soil parameters that we need is the bulk density. So because we have a sample of known volume, we can weigh that and work out the bulk density. Could you write on that, Maria? Oh yeah, you can have a pen. Fantastic. So that should be 15 to 20. What happens is when you use a screw extruder like this, you often sort of compress the core slightly. So the other core is actually a split core, and you bang it in from the top, but you open it so that you don't have to compress the core to push the core out. So that that organic layer is pretty thin. It's only a few centimeters thick, but it's really important in terms of long-term cycling of some of the radionuclides that we're interested in. And we probably would expect to find most of the cesium, plutonium, strontium. The strontium may have gone a little bit deeper, but the strontium will complex with organic matter and will get bound up in that sort of organic layer. Okay, so that's the soil sampling. The most striking thing is the wildlife situation. It really is quite an unexpected outcome of such a major nuclear disaster. It's quite contentious. If you say to anybody, actually, perhaps the long-term outcome of the nuclear disaster, at least in the immediate area surrounding the reactor, by that I mean the 30 kilometre zone, it's quite contentious if you say, well, actually, there may be a long-term benefit to wildlife. It's actually quite difficult to get that message across to people who haven't actually come to see the zone for themselves. When I was here 20 odd years ago, people were just starting to talk about the zone becoming a wildlife reserve, and at that time, I think I saw one moose back then. In the course of 2 days fieldwork, I've seen 3 moose and several deer and other animals as well, lots of birds. And we're hearing from the people, like the tree partners who are studying the wildlife within the zone. We're hearing that actually the wildlife, the biodiversity of wildlife, is really tremendously rich compared even with neighbouring areas. So it's a difficult message to get across to people. There's by no means the message should be that nuclear accidents are desirable or good in the long term generally, but I think it is a striking feature of the zone that that's what we're seeing.